The next speaker is Rick Bushman. He's the W.M. Measy Professor and Chair of Microbiology at the med school here at Penn. His research centers on genomics of host microbe interactions with specific projects on now coronaviruses, more traditionally HIV, the human microbiome, envirome, and human genome modifications. He is co-director of the Penn Center for Research on Coronaviruses and Other Emerging Pathogens, as well as a founding principal investigator of the Penn CHOP microbiome program. And he's going to talk about his most recent studies on waves of infection by SARS-CoV-2 variants in the Delaware River Valley. So, Rick, take it away.
Thank you, Susan. Can you hear me okay?
Yes.
Okay, great. So what I'd like to cover in this talk is some introduction on viral whole genome sequencing, SARS-CoV-2, and what we've been able to learn in our area. Then waves of infection in the Delaware Valley analyzed by whole genome sequencing. What are the variants that are circulating in our area? How have they changed? And what inferences can we make based on knowing more about it? I'll talk about the COVID-safe program, which provided some of our samples that we've used for viral whole genome sequencing along the way. And then the important question of are different variants more prone to breaking through with vaccination? What can we learn about that, given a rich background of understanding the virus in our area? And then if I haven't run out of time, briefly I'll mention some speculations on what's next. So this work was supported in part by the Penn Center for Research on Coronaviruses and Other Emerging Pathogens. Susan Weiss and I are the co-PIs. Our goals are to expand and accelerate SARS-CoV-2 research at Penn, CHOP, and Wistar, provide centralized information on SARS-CoV-2, and compile information and catalyze opportunities for new funding for research. And so here at Penn, we have a vast research effort on coronavirus, SARS-CoV-2, involving more than 100 labs. Penn, as you know, provides a unique opportunity to fuse basic and clinical research with the two types of research all pushed up together on a single campus. And already we've seen a series of research breakthroughs on COVID here at Penn, some of them helped along in part by the efforts of the Center for Research on Coronaviruses and Other Emerging Pathogens. So if you want to know what the latest is, please follow us on Twitter. We try to be active and tell you about what we're up to. So understanding viral evolution in the Delaware Valley. So the core of this work has been longitudinal surveillance sequencing, where we've generated more than 3,500 sequences of local viral genomes over time. And so that provides a rich picture of what kind of viruses are here, how they're changing. And from that, we can launch a bunch of other research projects interrogating this data set. So this has been the contribution from a series of multiply talented teams, a dream team of dream teams, the Penn Genome Sequencing and Analysis effort, led by Andrew Marks, Scott Sherelmix, John Everett. Our first sets of samples were provided from hospitalized patients from Ron Coleman and his team. More recently, Brendan Kelly has established a CDC-funded surveillance effort with Kyle Rodino, collection of collaborators, including the City Department of Health, the CDC, and others. That's been the basis of more recent work. The COVID-safe screening program has helped keep the medical school safe with surveillance testing of people, asymptomatic people in the medical school. And that's work of Arupa Ganguly, Scott Sherelmix, John Epstein, and a large team. A number of groups now are starting in on modeling the virological, epidemiological, and social aspects of the epidemic in Philadelphia. And this includes Carolyn Canusio and Katie Strello, Rachel Furstein-Simon, and then several other groups as well, Jeffrey Morris, Kevin Ricks, Doug Wiebe, and others. We have additional partners for carrying out sequencing in our area and contributed some of the genomes in that number, including folks at Jefferson and CHOP. And as we identify interesting viral variants, they're flowing back into the lab with work in Susan Weiss's group and others. John Wary, Mike Betts, Scott Hensley, Paul Bates have all been analyzing local viruses and trying to help us understand when we see some novelty in a viral variant, what are the functional consequences. And so in our first study, we analyzed viral genomes early in the epidemic, SARS-CoV-2 genomic variation in space and time in hospitalized patients in Philadelphia, work led by John Everett, Pasha Hukama, and all this work that I'll be describing is a collaboration with Ron Coleman. And in this first study, his team sampled hospitalized patients and provided the specimens we were able to study. And so here's the viral genome. It's about 30,000 bases, and we're determining the sequence of every one of those and then using that as a basis for making inferences about the virology. So in this first study, we sequenced 52 genomes from 27 hospitalized subjects, March through July of 2020, just as the epidemic was starting. And we compared those to global strains circulating around the world, and found that the strains we saw in Philly had closest neighbors in New York. So New York is a larger city, an earlier peak of COVID infection in the first wave. So it seems like in Philly, infection got launched as spillover primarily from New York. However, when strains from Philly were compared to each other, most had nearest neighbors within the Philly data set, indicating community spread within Philadelphia. And so in some cases, we had multiple samples from the same subjects over time or different body sites within the same subject. And in some of those cases, we're able to see real changes in the virus longitudinally or spatially. And so that said something about the ease with which this virus can evolve over time. And that was disturbing. And unfortunately, that's kind of a theme of the parts that are going to follow. So one thing we were able to see, was a characteristic change in the viral spike protein in this early data set. So the viral spike protein is this viral surface protein. It's very important for binding to host cell receptors and programming entry of the virus into human cells. This change, substituting a D aspartic acid for 614 at position 614 for a glycine G, was seen very early in the epidemic. The Wuhan strain had D, but most later viruses had G. And this just shows over time the replacement of this D version with the G version in early 2020. In the sequence, and so this probably promotes spike function, improves the ability of the virus to infect human cells, unfortunately. In Philly, all the viruses we saw have been this D614G type, indicating that this wave of viral evolution had already happened by the time the viruses got to us early on in the epidemic. So fast forwarding, we've had a series of new sources of samples for the whole genome sequencing, one being from the COVID-SAFE program. I'll just highlight that briefly. In this context, they're sources of specimens from asymptomatic subjects, but this was set up as a rapid assay lab early in the epidemic. As you well know, tests were not available when we needed them. So at Penn Medicine, we set up our own. Greg Van Dyne in biophysics purified enzymes, and we set up a loop-mediated isothermal amplification detection system. Outreach via cell phone apps, CLIA approved. With this home-brewed assay, Arupa Ganguly and her team screened no more than 70,000 specimens for folks at the medical school, checking that people weren't infected, and once in a while catching an asymptomatic carrier and providing them feedback so they could sequester and not infect a lot of other people. And that also provided a source of samples for genome sequencing from asymptomatic subjects. And just to tell you a little more about this effort, Scott Sherrill-Mix in our lab developed a improved detection method that was used for the 70,000 specimens. This RT lamp method is a little messy. It's a little too easy to get amplification, so there's high background in the assays. So Scott developed this molecular beacon detection system. So what a molecular beacon is, it's kind of cool. It's a DNA molecule, this loop here, where the ends of the DNA are such that they anneal with one another. They bind to each other. There's a fluorescent group and a quencher on each end. So the fluorescence is quenched by formation of this hairpin. But you've designed the molecule so this internal bit can bind to SARS-CoV-2 nucleic acid, which you're trying to detect. And so when it binds, that separates the fluor from the quencher, and you get fluorescence as a result. And so this was the core assay we used for this large screening program. And it took some very clever engineering on Scott's part to get this to work right. You had to do the reaction at certain temperatures. And so to get this binding to work at just the right way at the required temperatures, Scott introduced some locked nucleic acids, some non-biological nucleic acids that bind more tightly. That's the little red loops here. And that was a required part of the design to get it to work really well. And again, that was used to screen more than 70,000 specimens. So fast forwarding to today, here's what the picture looks like. We've been sequencing and displaying what we find on a dashboard of the variants in our area. You can go online and check this out whenever you want to see what variants are present in the Delaware Valley. So the x-axis here shows time. The y-axis shows the proportion of different variants. And these are samples we've collected from a bunch of different locations. Primarily the pathology and lab medicine, Kyle Rodino, Brendan Kelly, I'll say more about their efforts. The COVID safe group, Ron Coleman's sampling of hospitalized patients, colleagues, samples from the Jefferson catchment, CHOP, and also other sources. And so here's the summary. You can see we've seen waves of different variants as indicated by the different colors. Early on, it was the B1 lineages. Then this green is B1.1.7, which is the alpha lineage, the lineage first detected in the UK. And then that was displaced by these red and brown lineages, which are all delta. So today, everything that we're seeing is delta in one form or another. So this just, here's an analysis from Andrew Marks, who's done a lot of this, led a lot of this sequencing effort recently. A sort of artistic way of viewing the progression of the epidemic. Here he's done a pairwise comparison of all of our genomes, each to every other one. So this summarizes 9 million comparisons. And over here, he scored the diversity, how different the genomes are from each other. Whereas if they're identical, they're this dark blue. And as they get more substitutions, they get redder and redder. And so it's time going this way, and all against all comparison. And you can see that early on in the epidemic, there was very little diversity. The virus had just arrived. It hadn't changed much. But then over time, you can see the diversification as it gets more red. And then a little June 2021 or a little later, suddenly much lower diversity. And this is the alpha wave. We were seeded with alpha. It quickly took over, displaced everything else. And now it's all alpha. Here's another way, just comparing the clinical picture a little more with the waves of infection. Again, time on the x-axis, percent of the total on the y-axis. Green shows the alpha wave, red shows the delta wave, and we can see sequential waves of hospitalizations in the dark gray and test positivity in the light gray. And you can see peaks associated with the alpha wave and then with the delta wave. The waves of infection have been similar but not identical nationally. So here's another display from Andrew comparing the longitudinal patterns of variant accumulation in different cities in the United States over time. So here's Philly, as I've been showing you. Here's New York City, Washington, Phoenix, Los Angeles, Seattle, Chicago. You can see that everybody has this wave of alpha and then this wave of delta. But some of the other variants that have appeared differ. So for example, over here in Los Angeles, you can see this epsilon or light yellow variant is quite prominent. A little later, it's prominent in Seattle, not quite as prominent. Chicago, some, but not so much on the East Coast. Whereas on the East Coast, we see this wave of iota, this light purple from New York. We can see some, but less in Philly, and proceeding south, some, but even less in DC. So there's sort of a north-south gradient of the appearance of iota. So ongoing change and some geographical differences. So what about vaccine breakthroughs? An important question is, are any of the variants more dangerous to us? And particularly, are any of them more prone to breaking through, infecting vaccinated people? So we've just submitted a paper from this large team. This is taking advantage of primarily of the clinical sampling led by Kyle Rodino and the CDC project, organized and funded by Brendan Kelly. So it's actually a complicated question to ask if any variants are more likely to break through. So down here you can see we're color coding again the variants, and these are all vaccine breakthroughs. And we have some numbers over some periods, but you have to compare that to the changing background of variants to ask if any of them are enriched. Are we seeing them more down here than we do up here? And so it's complicated because there's this stochastic sampling. You can see it's sort of choppy, and the background is changing over time. So again, Scott Sherrill-Mix developed an advanced statistical treatment, Bayesian autoregressive moving average logistic multinomial regression, that allowed a rigorous comparison. And so first he had to smooth out the rough data, assuming that last week is going to be like this week, and similar to next week, there may be a trend up, trend down, but you expect a smooth progression, and so he smoothed the data, so this is probably closer to truth. And then each week is compared to the appropriate time in the smoothed background, so that we take account of the fact that the background itself is changing. And so carrying out that analysis allows detection of, in fact, some of the delta lineages do seem to be breaking through more efficiently than other lineages. So particularly, here's this one delta lineage B.1.617.2. This shows an odds ratio where if there was no enrichment or depletion of vaccine breakthroughs of this lineage, then the peak would be at 1, but the probability density, as you can see, is moved to the right. So the odds ratio is about threefold enriched, or about threefold more likely to have a delta breakthrough than for other lineages. Similarly, some of the other deltas show enrichment and this P1 lineage, whereas some other lineages show that they're less likely. So unfortunately, delta looks a little more prone to being able to support vaccine breakthroughs than some of the older lineages. What about point substitutions? What about just changes in the virus independent of what variant they're in? So Andrew and Scott did an analysis of this. So here's again time on the x-axis, proportion of genomes on the y-axis, and these are a bunch of point substitutions, green being ones that are seen in alpha, orange ones that are seen in delta. And again, we carried out the sort of advanced statistical analysis, and this one spike substitution, N501Y, is actually enriched, and about a twofold increase in odds ratio. This is a very well-studied substitution in spike. It's nicknamed the Nelly mutation. It has been reported, it's found in the alpha, beta, and gamma variants. It's reported to increase binding of the spike protein, the external viral protein, to ACE2. It may reduce antibody binding, and it's been suggested it may be a keystone mutation, that once you get this substitution, the Nelly substitution, it's easy to get a lot of other substitutions which optimize viral replication in humans. That's sort of disturbing to see a relatively common viral variant that seems to be associated with vaccine breakthrough. We could see others that are a little less prominently associated. Here's one in the spike that promotes the furin cleavage, which is important for infection. I'll say more about that in a minute. And then other substitutions were not enriched, or some of the other substitutions that have been closely studied and seem to improve spike binding or antibody evasion were not changed, like the EEC484K substitution did not seem to be enriched in breakthroughs in this data set. So another way of seeing ongoing evolution of the virus in our area for infection in humans is the evolution of composite variants. So the alpha variant, the variant first seen in the UK, for example, has appeared with this additional spike substitution, the EEC substitution, which blocks binding of certain antibodies and promotes binding to the ACE2 receptor. Paul Planet's group noticed that this was circulating in the Philadelphia area. Also, this other one, 21 from Columbia has the EEC substitution, the Nelly substitution, and others that are important for infection in humans, all piled up on one genome. There are other composite variants that we haven't detected yet. The AY 4.2 or Delta Plus has been much in the news lately, and we've not yet seen that one, and others as well. So there are multiple examples of variants of interest or concern acquiring further notable spike substitutions, and this supports the broader picture that SARS-CoV-2 is evolving to grow and spread more efficiently in humans based on data in our area and globally. So there's been a lot of interest in bringing this quantitative analysis of variants together with lots of other kinds of data to understand viral spread, the role of healthcare disparities, and the effects in our area. So this map shows some work from Katie Strello, where she's analyzing the numbers of cases per 100,000 residents, here projected on a map of Philadelphia. And the pie charts show the different variants that are present, alpha, delta, and others. And so you can see the alpha taking off, then delta, and you can see regional variations in number of cases and the specific variants that are present. And so she and a number of other groups are interested in trying to model the spread of the virus locally based on the type of variants, the extent of vaccination, infection rates, social vulnerability indexes, other measures of disparities to try to understand why the virus is spreading where it is, project where it might go, and devise optimized interventions. So lastly, as we obtain new strains from our area, we're funneling them back to the lab, and Susan's lab is studying how they grow, how they differ from each other, and trying to make inferences that might be important in understanding mechanisms of viral spread or its inhibition. So just to show you a couple of kinds of assays, the spike protein, as I mentioned, has to be cleaved. It has to be proteolyzed, broken into smaller fragments in order to work, to allow entry into cells. And so here her lab has studied several of the variants, and you can see alpha and delta have this more efficient cleavage, producing this lower band, the S2 subunit. So that is likely part of the reason that they grow efficiently. Also, these lineages have changes near the site of furin cleavage, one of the protease cleavages, P681H or P681R, which may well be at least part of the reason they're cleaved more efficiently. Here, this is again from Susan's lab, from Nikki and Anant. Local viral isolates can differ from each other. So these are photos of cell culture dishes where here are the cells just growing. Here, if you had SARS-CoV-2, you can see the cells round up, don't look as good. This is called a cytopathic effect. And here, Susan and her team are studying a bunch of local variants that Ron's team isolated, Paul Planet and CHOP. And you can see that morphologically, some of them look different from each other. So this is now beginning to develop lab-based distinctions among the different lineages that then provide the basis for trying to understand what's the molecular reason and what does that tell us about how the virus is growing, and can we use that to our advantage. So I'll stop there and summarize. We've determined high-quality sequences for more than 3,500 SARS-CoV-2 genomes from the Delaware Valley. We've seen sequential waves of infection, the B1, D614G, then alpha, then delta. Vaccines are highly effective, but breakthroughs are enriched when you're infected with a B1 617.2 lineage, emphasizing the high infectivity of delta. Vaccine breakthroughs are also enriched in the presence of the point change, the Nelly mutation, N501Y, which may be a foundational mutation for a collection of further changes that promote replication in humans. We've detected composite variants like B117, the UK variant, alpha with E484K, the B621, and others, consistent with ongoing evolution for efficient replication in humans. And local variants can differ from each other in careful mechanistic assays. So briefly, to speculate on where it's all going, on the downside, we're seeing viral spread between vaccinated individuals. And unfortunately, this is a setting for evolution of the virus to elude immune responses and spread even more efficiently between humans. How much the virus can change, how much change it can sustain and still work properly is a big question. Probably it's less plastic than flu. So maybe that's good news, but we'll see. We're seeing seeding of new animal reservoirs, unfortunately, in cats, dogs, minks, white-tailed deer, and various zoo animals. So this is potential for the virus to spill into new creatures, evolve there, spill back into humans. And of course, there's very widespread global infection, particularly in vulnerable populations and spotty vaccinations. So unfortunately, I think that means we're probably going to see circulation of SARS-CoV-2 in the human population indefinitely. It's hard to see how it's going to go away permanently. However, on the upside, vaccination is highly effective. It suppresses spread and greatly mitigates severity. So get vaccinated if you haven't been already. It's even more effective with boosters. We're now seeing orally available small molecule therapies. So I'm hopeful that even if COVID is widespread, the impact will be diminished. But of course, that means efficient implementation of vaccination and therapy over all groups who need it. So I'll stop there. Thanks to all the collaborating teams, again, the dream team of dream teams, and I'll stop and take questions. Thank you very much.
Thank you. Thank you, Rick. Can you hear me?
I can hear you.
That was really interesting. I have a comment and a question, and then there are a couple of other questions. So the N501Y mutation that makes it do better or interact better with the human receptor, it's also one of the amino acids that aids adaptation to mice. So that's important for the mouse-adapted SARS strain. So I'm thinking that mutation may just make it more broad tropic, may help it, to spread to all these animals. But my question is, the E484K mutation, the so-called EEK mutation, which is also, that's the one that's supposed to be kind of an immune escape kind of mutation or aid an immune escape. It's interesting that mutation is not particularly associated with the Delta breakthrough. So does that suggest the Delta breakthrough it's not really an immune breakthrough, but just because it's such a functional virus? What do you think about that?
We're not seeing that one on Delta a lot. So,
right.
Yeah, I don't know. Is it incompatible with the other Delta substitutions? Does that make a bad virus, or is it just redundant? What's already in Delta does the same thing as well or better? I don't know why we haven't seen that in the Delta background. That's a good question.
But what I think is interesting about that is that the Delta is the one that's doing the most breakthrough in vaccines compared to the others, but it doesn't have the mutation that is reported to be the immune escape one. So the vaccine breakthrough may not really be related to immune escape as much as just a better virus, a more adapted virus.
I think that's very possible. The Delta seems to grow to higher higher copy number, more RNA copies, lower CT in the PCR assays, which may just say something about robustness of growth in human cells rather than anything specific about immune escape. Yeah.
And that's the one that's easiest for us to isolate from the patient samples, because of the low CT. Mm-hmm. Okay, so here's another question. Excellent presentation, Rick, from Liz Anderson. For the vaccine breakthrough analysis, with Delta having a higher OR, I'm not sure what OR means, but breakthrough. Oh, okay. Have you looked at time since vaccination to see if that is an impact?
That's a very good question. We would love to look at that. Unfortunately, in order to be able to access large numbers of clinical samples, we have to forego getting a lot of the clinical data. So we don't have that information. Similarly, we don't know if the vaccinations worked. We don't have data on immune responses. We're trying to set up a project now to get that data and compare variants and breakthroughs under circumstances where we measured if the effectiveness of the vaccination procedure itself. So we're relying on large numbers, and our metadata is unfortunately thin because of the regulatory background.
Here's another related vaccine question. Can you tell if the rates of viral mutation are mitigated by high vaccine rates? That is, fewer patients in which to replicate, leading to fewer opportunities for virus to mutate.
We've not seen that, but that's a very good point. It would be interesting to know if vaccination means that virus grows to less high levels, there's less total virus, less opportunity for changes to accumulate. We've not measured that, but it would be interesting to investigate. I agree.
I think also, just generally speaking, the reason we get changes is because the virus replicates. So there's a combination of it replicating less with vaccination, but also it may, as Rick mentioned, it may be the mutations may be asymptotic. It may be there's only so many that the spike can tolerate. So it might be hard to separate those two factors. Okay, this one might be good, more for me than for Rick, but let's see if Rick can answer it. Due to its proofreading enzyme, SARS-CoV should hypothetically be less liable to mutate rapidly than similar viruses. Does the high diversity wave indicate this to be false, or is it simply a matter of having so many hosts that even really low mutation rate will result in this diversity? From Tzvi Pollock.
I think it's the high number of hosts and just the raw amount of total virus. It is a little more accurate in replication than some other RNA viruses, but there's just so much total that there's a lot of opportunity for new variants to arise. One thing that might be worth mentioning, we're accustomed to the idea that flu changes a lot, but flu, the virus is in segments, the genome is in pieces. And you can reassort pieces and get a really different flu due to antigenic shift, as it's called. And there's nothing like that in coronaviruses. They have a single long RNA chain. So there is this important mechanism that's present in flu is not present in coronaviruses. So that's good news.
I mean, the coronavirus proofreading enzyme reportedly reduces the error rate by about 10-fold. So it's still a pretty high error rate. And the genome is so long that, it's just a larger target for so-called mistakes or mutations. So it does mitigate a little bit the rate of mutation, but but like Rick said, not enough to catch up to this incredible numbers of replication cycles. Okay, we got lots of questions here. Liz Heller, thank you, Dr. Bushman, for an excellent talk. Can you tell yet if rates of viral mutation are mitigated? Oh, by high vaccine, that's the same thing. It's pretty much the same question. Okay, great talk. Adding to E484K, P1 and PB13.351 that also have the E484K were not that successful to spread globally as alpha or delta. It's sort of what I was saying, I think, right? You can address that.
So why is that? Yeah, I think it's really interesting. We tend to focus on these like glamorous spike substitutions, the EEC mutation, Nelly, and so on. But there's plenty of other substitutions all through the genome, and most of which are unstudied for their effects. And so I think there's a great deal more to learn about how different substitutions in the viral genome are affecting replication and spread in humans. Susan, do you want to add?
I mean, that was one of my questions. I was going to ask you that question too. And I think I know the answer because we've talked about it a little bit. There probably are going to be mutations outside of spike that may be important for spread. I don't know that anyone's really looked at those carefully. I don't know if your group has really done that.
There's starting to be a little bit of work, sort of the mass spec fishing, looking for host cell binders and does it change the binding. Also, some of them may be epitopes for cellular immunity, and so there are some programs that can try to pick out good epitopes for cellular immunity, but that's extremely complicated because humans differ from each other in HLA, and so they'll respond differently to different changes. So it's a very hard problem, and I think people are starting to scratch the surface, but my guess is there's a lot more there.
I personally think somebody should really take a look at the other gene. That's a little bit different approach from what you're talking about, but to just do the sequence analysis of, say, the replicase, or the innate immune, the immune evasion genes, because any of those could change virulence as well as spread. And nobody's really looking at those, as far as I can tell. All the attention's been on spike, which is clearly the most important for spread. Okay, here's a good question for you. I don't know you'll know the answer to this one. Patrick Cherubin, based on downside remark you made, is there still some hope to reach herd immunity sometime soon?
Well, herd immunity, the idea kind of goes away if the virus is spreading in vaccinated people or people who've already been infected. The virus will continue whether we're immune or not. So I don't know. We don't seem to be getting complete sterilizing immunity. But that said, there's a lot less viral replication. It's a lot less pathogenic. You're much, much better off if you're vaccinated. But it's not like that's making the virus completely go away 100%. So I guess I'm not seeing sort of population sterilizing immunity likely to happen anytime soon. On the other hand, my predictions about many of these things have been completely wrong. So I hope I'm wrong on this one, and we can make it go away, but I guess I'd be surprised.
Well, we might have herd immunity against disease, not against the virus, at least in our environment, not everywhere in the world. Okay, I think that's about all the questions. And thank you so much, Rick. That was really, really fun, interesting.